very good. But they're gonna, you might have seen drawings like this before, many different spots where replication is beginning and then moving in both directions. If they didn't start at many different spots, it would just take too long to replicate all the DNA. Okay, so prokaryotes have one, eukaryotes would have many origins, many beginnings of replication. And I will mention that replication proceeds in both directions. In 2 directions. So it's, this picture is not my favorite. It's from your book here, figure 7.7, because the color of the old strand is so close to the color of the new strand that it's hard to miss, easy to miss what's going on. So here's the original DNA. It's kind of a dark blue. And then we have the helicase pulling the 2 strands apart. That's happened here. And then replication, again, it's going in both directions. We have 2 replication forks. We're going to look more at that in a minute. That proceeds both ways. Sometimes this is called a theta structure because it looks like a Greek theta. And then eventually all the DNA is replicated. And again, you have another copy of the DNA. Okay, and then that always is going to happen before a bacterial cell splits and before it divides. Okay. All right, let's look at, there's 5 or 6 enzymes that I want you to understand their role in replication. Okay, and the star of the show here is going to be DNA polymerase 3. There's actually 5 different DNA polymerases, and we named them in the order that we discovered them, apparently. But DNA polymerase 3 is the one that makes the polymerization reaction. It adds those triphosphate nucleotides on to the existing strand. Let's see. All right, so let's write that. That's what the enzyme does, adds triphosphates nucleotides. To the 3' end of the growing DNA strand. Okay, so for some reason this copied weird. This is supposed to say 3' end, and this is the 5' end. So the new DNA, and this actually shows a little bit better in the next graphic. It's going to be going in this direction on this strand because this is going to be the 5' end of this strand and the 3' end here. Remember we said those nucleotides are always added on the 3' hydroxyl group. And this bottom strand is going to have to go this direction because, again, this is going to be the 3' of this strand and 5' of this strand. Okay, so let's see a little bit more about that enzyme. It works pretty quick. Adds 500 to 1,000 nucleotides per second. All right, so a fast polymerization reaction. It's also capable of doing some proofreading. So it actually can add the nucleotide, and then it kind of looks behind it to make sure the last one it added was the correct nucleotide. So in other words, it checks for mismatch errors, adding the wrong base, maybe one that wasn't complementary to the template. So it has a proofreading exonuclease function, because that's really important to copy it correctly. You're making the blueprint for cells. If the blueprint is wrong, then every protein that eventually gets translated is going to be wrong. So it's important to have the instructions correct. With that proofreading function and some other error fixing mechanisms, we have about a 1 in 10 billion error rate, and that's in mismatch. Okay, so that means, again, putting the wrong base that's not, say, putting a T with a G or something. They're not complementary bases. All right, so a pretty low error rate. Well, that's good. That's what we want. All right, what other enzymes are doing work here? Well, it turns out that RNA polymerase, it's a big enzyme and it does a lot, but it can't just it can't just add nucleotides, we call it de novo, from nothing. It can't just start on its own. It has to have a 3' hydroxyl group to start out with. So another enzyme has to come in called primase that adds an RNA primer. So a short 10 to 20 base pair, okay, adds 10 to 20 base pairs of RNA to give DNA polymerase 3 a 3' hydroxyl group to start. Now, keep in mind, this RNA that it adds is temporary. Okay, it's RNA. You can't have a little strand of RNA in your DNA. So that's just temporary. Other enzymes are going to come in and cut that out and put nucleotides in there and not ribonucleotides. Okay, so primase does its work here. This shows, again, this is the leading strand. We'll get to the lagging strand in a minute, adding those triphosphate nucleotides. And again, this is going to be the 3' end of that new strand. Okay, the lagging strands. I think we have time to go through this. All right. Oh, let me point out here, this enzyme right here, this is the helicase. Okay, that's the unzipping enzyme. And then there's going to be some stabilizing proteins here that just keep that replication fork open. Okay. All right, the other enzymes involved here are mostly with the lagging strand. All right, so in the other strand, again, it has to work in short pieces away from the replication fork. And those short fragments are called Okazaki fragments. Somewhere between 1,000 and 2,000 base pairs long. Okay, so you're going to have a series of primers. Okay, they're in red here, here, and here. The RNA polymerase then replicates that DNA in strips. Okay, and then it'll come back here and do the next one and come back and do the next one like this. Those RNA primers actually are excised by an enzyme called RNA polymerase 1. So it gets rid of primers. And then fills that in with DNA. And then there has to be one other enzyme that comes in, ligase. Sorry, I'm trying to do this fast. Ligase basically seals the ends. I think actually what it does is add a phosphate. So just to make sure that that DNA is all one strand. Okay. So the important enzymes there, RNA polymerase 3, the helicase, the stabilizing proteins, RNA polymerase 1, ligase. Okay. You're going to want to know what those all do, the directionality of it. There is a nice YouTube video type cartoon, it's just a couple minutes long, that uses all those terms and just kind of shows you an animation of how that works. And it's just very interesting to kind of see it in, real time. Yeah.
Those stabilizing proteins, isn't that gyrates?
Ah, the stabilizing proteins, aren't those gyrase? No, the gyrase has another job that I will start with next time. So I'm glad you're thinking about gyrase, but we haven't come to that yet. Okay, have a good day.